Okay, so for this video I want to show you how to do an old-fashioned style genetic cross. This is how we'd set it out without a Punnett square. You get quite a few exam questions that can look like this and also in old textbooks, so it's really helpful to know this way of doing it, even if you rarely use this way of doing it. You can only do it for a monohybrid cross. You can't do it for anything more complicated than that. So what we do is down the side of your page, in the margin if you like, we're going to write ourselves some little headings. First thing we write down is what the parental phenotype is. Okay, and then below that we write down on the next line the parental genotype. Alright, so for the one that we're going to look at, we're going to do cystic fibrosis. Let's do something that you're really familiar with. The parental phenotype, we're going to say both parents are going to be healthy, but they are also carrier parents. Okay, and we're going to have two parents who are both healthy carriers. Okay, and their genotype, because of that, is going to be big F, little f. Both parents, big F, little f. Okay, the next row down is where we're going to write down what the gametes are. Now this is the bit that really helps you with other more difficult questions, okay, being able to work out what the gametes are. Now here it should be pretty obvious that when this person produces gametes, every sperm cell or every egg cell, we don't know if this is the mum or the dad, is going to either have a big F or a little f. So that's what we're going to write. To show that they're gametes, we draw them in a circle. So a big F in a circle. Okay? Or we have a little f in a circle. So we're indicating that these are inside cells. Okay? The other parent has the exact same genotype, so they have the exact same gametes available to them to pass on. A big F in a circle and a little f in a circle. Okay? Now, this is what we're going to do next. We're going to leave ourselves 3 lines down the side. Okay? And what we're going to do is just on the line above where we're going to write into our next space, we're going to put some dots along. So what I'm going to do first of all is I'm going to say what our next row heading is going to be. We want to see the offspring. Okay? So I'm going to write F1 genotype. Okay? F1 is the term given to the first offspring generation. It comes from the Latin word filius or filial, which means son, like son of, which is sexist, I know, but that's just how it always was done, so that's what they've stuck with. It turns up a lot in horticulture, so in plant growing for garden centres, things like that. You'll find bedding plants in little trays of 6, like a plastic tray with 6 identical plants in it, or polystyrene trays. And it will quite often say on the label that it's an F1 hybrid. That means that they've crossed 2 plants where they know the genotype, and they know therefore what the colour of the flowers will be, what the height of the plant will be. It means that they have known offspring because they have known parents. So that's the only place really you're ever likely to come across that in real life. You will see F1 hybrid written on a lot of plant sales material. Okay, so what we're going to do is imagine that you've got your line here, which is the line above where you've got F1 genotype, and we're going to put a dot below each of my circles. I'm going to have a dot. Okay, this is going to help me when I do my next step. So for the next step, what we want to see is, well, what are the genotypes that we're going to get if we cross these parents? And we're doing it without using a Punnett square. The row after that is going to be the F1 phenotype, which is going to tell us the characteristics based on those genotypes. Okay, and then the final row we're going to have is the ratios. So, for the F1 genotype, first of all I'm going to look at what's going to happen if this gamete here combines with the first gamete from the other parent. Obviously we can't cross these two together because they're from the same parent. Has to be a gamete from one parent with a gamete from the other parent. So I'm going to do it in order, and I'm going to have those two together. Okay? Can you see that that is going to give us a genotype of big F, big F? Yeah? Then I'm going to do the next one. So I could still use this same gamete again because I've only used it with this one. I haven't used it yet with this one. So my next step is to go, well, that same gamete, but with the other one from the other parent. Okay? If you always set it out like this, you will never ever go wrong with these. This one's going to give us the big F that we had before, but now I'm combining it with a recessive allele. I've got a little f. So I've used up this gamete. There's nothing else I can combine it with. It can't combine with that one because it's from the same parent, and we've combined it with each of these two. So that gamete's done. Now we can move on to this one, and we can say, well, what will happen if this one combines, first of all, with this one, and we write down a dominant allele first. So we don't write little f, big F. We write big F, little f, just a standard protocol how you write it. And then can you see again, this one's not been completely used yet. I've only tried it with this one. I've not tried it with this one. So that's going to be my last cross, is that combination, which is going to give me my homozygous recessive combination, isn't it? Okay? And I don't draw these in circles because these aren't gametes. Gametes have only got one allele in for each of the genes that we're looking at. We're only looking at one gene, the CF gene. So then I can look at the phenotypes, and I can see that actually all three of these are going to be healthy. Yeah, healthy from CF. Yeah, they've not got the disorder. And this one here is our sufferer. Okay, and then the ratios therefore are going to be 3 healthy to 1 sufferer, and that's how we write it out. The way you can tell if you've done this one right is that every gamete should have 2 lines coming out of it. Okay, so this one has got 2 lines, this one's got 2 lines, this one's got 2 lines, this one's got 2 lines. I've done it right. If any of your gametes ever end up with just 1 or 3 and all the others have got the right numbers, then you obviously realise you've gone wrong somewhere. But if you always do this one with this, and then this one with this, and then do this one with this, and then this one with this, always do it in the same format. You can tie yourself in knots trying to do it in a random order, so it's much, much easier to always go about it in the exact same way, use the same structure every time you do it. You'll get it right every single time. If they'd wanted to know the ratio of carriers, then obviously we'd have split this out and said healthy, not carrier, healthy carriers and sufferer, and therefore we'd have had a ratio of 2 because these two would be our carriers. Okay?